So today I'm going to tell you about the applications of microfluidics in quantitative biology. There are three parts in my presentation: overview, microfluidics devices, and the last one, conclusion. First of all, you have to know microfluidics is kind of a technique that can manipulate fluid, collect data in high throughput as well as quantitative manner. Move on to classification of microfluidic devices. There are two major types. It depends on the location of cells, channel paste, and product paste. You can look at the picture A and picture B, and picture A describes the difference between traditional devices and microfluidic devices. There are 2 points you have to focus. This is the equipment they used, and the second is amount of medium it used to cell culture. And the picture B describes the way that microfluidics promote the development of quantitative biology. There are each devices. There are 3 typical features you can look at in the pictures, and more importantly, you focus on requirement with regard to quantitative biology, such as analysis, measurement, and environmental control. Yeah, let's to talk about to be more specific. The first devices is channel-based microfluidics. There are 3 typical features: flexible manipulation, high resolution, and the last one, redefined function. And let's talk to overview of the features of channel-based microfluidics. It enables fluidics manipulation, such as some action like flowing, mixing, confinement, as well as chemical reaction. It also cell culturing in a channel or chamber on micro scale. Other than that, it also provides a well-defined and long-term living environment for real-time observation. And thanks to the significant point, researchers can be capture the change of cell behavior for the purpose of quantitative analysis. The first typical features is high resolution. Each need to use a small volume of growth medium, but it's also long-term culture. It can so much for high resolution observation and real-time observation. And Europe depends on the feature to co-culture to synthetic strain and spray and rather in a micro chemostat chip. And the result, they observe extinction, coexistence, as well as oscillatory dynamic of the ray radiator system under different conditions. And the second typical feature is flexible manipulation. It includes diffusion, velocity adjustment, and medium switching. You have to know the roles of flexible manipulation. There are a lot of benefits, such as define stimulant, control environmental changes, as well as quantify the corresponding responses. With regard to instrument, you have to know there are 2 kinds of instrument. This is internal and external, but it have a common function. It control the flow rate of fluid, uh, precisely. Um, and I take a theory of study to kinds of detailed example for flexible manipulation features. Theory design a devices combine between a microfluidics channel and micro valves, and the result they measure the quantitative relationship between phenotypic diversity and molecular performance of single bacterium in a chemostatic wave. And the last feature is redefined function. It's a be designed for various purposes. For example, on multicellular system like biofilms. And if you want to know more about a biofilm or how to create a biofilm, you have to search for further information later. I just demand this because the Salzburg used biofilm to observe, to apply his study, and he successfully observed an oscillating growth behavior of a monolayer bacterial biofilm in a chemostal microfluidic chamber. And let's move on to the second devices. This is probably paste microfluidics. You can look at the feature 3, describe the application of the droplet-based microfluidics in quantitative biology. And droplet microfluidics can generate a sample, water droplets, at an extremely high throughput with controllable size. And each droplet is surrounded by the sprouting orifice and is considered used as isolated reactor for a cell living inside, something like that. After that, when biocompatible polymer are involved, its droplets can be solidified and from a dependent three-dimensional environments for cell culturing. And if you could integrate the various method, you could be tested or analyzed or sorted to answer diverse questions in quantitative. The last to be more detailed and help you understand more about droplet-based microfluidics and the applications. Its applications, I will give you a video.
Microfluidics studies the behavior of fluids at the micro to nanometer scale. Microfluidics can miniaturize the work of a whole lab onto a microfluidic chip less than a few inches big. One thing microfluidics is great at is making consistent droplets of fluids. They can be used in cosmetics, chemistry, biology, and even as medicine. To make droplets the regular way, take a beaker filled partly with water and partly with oil, two liquids that can never mix. When you stir, droplets start to form. They are of different sizes. When using this technique at an industrial level, inconsistent droplet size can be an issue, and some of the liquid will be lost because you cannot control the quantities precisely.
Now, imagine being able to control the exact size and quantity of droplets with a faster and more precise process. Microfluidic equipment can do that. For the same experiment, you need a basic microfluidic chip, a see-through slate with engraved channels only a few nanometers wide.
In the microfluidic channels, you can control how you make the droplets. Put the fluid for droplets in one channel, the other fluid will pinch it from both sides and make tiny drops of fluid that collect in the pool on the right. Their size is strictly controlled. There is no waste of product.
No worries,
The process is fast. You can make up to 1,000 droplets per second. Great. But why make droplets at all? Well, this miniature
is process microcreated droplets.
It can be made of any type of fluid, and we can drop what we want to observe inside. This microreactor droplet can be used for many things. Cosmetic creams and lotions, supramagnetic nanoparticles, diagnostic tests and assays, mayonnaise and low-fat food with fat particles, controlled medicine delivery with biodegradable microparticles, and many more.
Certainly after you watching this video, you have to more understand how to create a droplet, and you also know how to put things inside a droplet to help you study. You have to apply for further study. I take a cell, for example, we can use the droplets as a carrier. So a cell is placed inside, and the droplets grows whenever we want, and the cell release. Other than that, droplets can be used as many feature dishes in which we can do cell culture, and lately we have even able to sort their droplets based on their color. And now let's move on to the second feature in isolated environment. As I told you before, because each droplet is surrounded by the outer oil, so it's protected from crosstalk and contamination. And other than that, it is considered as an isolated reaction. From that, its application is to perform complex biological reactions, quantify the target molecules, as well as use as tool to analyze the myths of cell-cell communication. And the last feature is three-dimensional transform. And I take because the features and how to create an independent three-dimensional environment for cell culturing. As you may know when you watch this video, and I also told before. So I trust the manual application of recent experiment. He cultured primary hepatocyte in microcapsule of liquid cord and polyethylene glial shell, and finally he realized the primary hepatocyte show higher production of albumin and other metabolites in three-dimensional microenvironment in more than two-dimensional cultures. And the last one is a move on to conclusion. You have to know the qualitative biology require precise environment control, quantitative measurement, and quantitative analysis. And Microfluidics sheds more light on quantitative biology from the ancient of microscale, fluidics manipulation, and precise control. And this is the whole my sentence today. And thank you so much for your listening.